Hi everyone, my name is Jakub. This is the next episode of MedChem Essentials, scientific seminars devoted to medicinal chemistry. Today we'll be talking about solubility assays. Solubility is the current topic of our discussions. Before we're talking about permeability. Today's presentation will begin with brief introduction to solubility assays, then description of thermodynamic solubility, kinetic solubility, and followed by scheme for application and selection of appropriate solubility assay, which will be followed by example data from thermodynamic solubility assay. Previously, we were answering the question why solubility is important. It is important because all compound molecules must be dissolved to undergo any pharmacological process. Next, we will discussing what determines solubility. But we missed one thing, and this is pH. As most of the drugs are ionizable molecules, their solubility will be affected by the pH of the environment. And this is the diagram for basic molecule. So in high pH, there will be a low solubility of this compound. When will be decreasing pH, increasing acidity of the environment, there will be increased concentration of the ionized form of the compound, which will be equal to the unionized concentration of the unionized form when the pH reaches pKa. When we'll be going down with the pH, the concentration of the ionized form will be increasing, and so does the solubility of the compound. And we'll get maximum solubility when we'll maximally ionize the compound, and all the unionized species will be completely ionized, in this case protonated. Let's move on. Solubility assays can be used applied for different reasons. One is to check which compound is most soluble in the dosing vehicle or in a solution, basically, or to select the best dosing vehicle for a compound. The dissolution rate of a compound may be increased by using different salt form, reduced solid particle size, or solid dispersion form. Now, before we'll be moving to description of different solubility assays, it's good to introduce the benchmark for solubility. What is the good or bad or acceptable solubility of a compound that we'll be measuring in our assays? Now, Lipinski used kinetic solubility assay for commercial drugs. He found out that 87% of them had solubility of at least 65 micrograms per milliliter, while only 7% had solubility below 20 microgram per milliliter. This was followed by a minimum acceptable dose approach applied by Kuratola and Lipinski. They found that the compound with a clinical dose of 1 milligram per kilogram and average permeability would require a solubility of 52 micrograms per milliliter for full dose absorption. So all this suggests that drug discovery scientists should aim for a solubility of a compound of greater than 60 micrograms per milliliter. Now let's move on. With this knowledge, let's move on to solubility assays themselves. We'll begin with thermodynamic solubility. And the shake flask method is a gold standard. It's very simple. We place a compound in the aqueous solution, the vial is capped, it has been shaken for 24 to 72 hours, then it is filtered, and concentration of the compound is measured by different methods. This can be UV, HPLC, UPLC, LCMS. Some assays can use specialized vials that have filter embedded within them. What are the pros and cons of thermodynamic solubility? It is the most advantageous assay for drug discovery, animal dosing, and preclinical development, because there compounds are dosed in solid form and they must dissolve completely, and thermodynamic solubility uses the compounds in solid form. However, the length of time from 24 to 72 hours makes the assay actually lower throughput than kinetic methods. So it tends to be used for smaller number of selected high-value compounds in later stages of the project. One importantly to note, to remember that unexpectedly low bioavailability of a compound may be caused by low solubility in the intestine. And in this case, this T-sol thermodynamic solubility assay can be performed using simulated physiological fluids. But we will discuss this on later MedChem Essentials. Now let's move to kinetic solubility. Here, the compound is added to aqueous buffer in a small volume of a concentrated DMS stock solution. Supposedly, the compound either is dissolved or precipitates. And there are like variations of the assay. We can either remove the precipitation or not, but either way, concentration, formation or not of the precipitation is being measured by methods that we'll describe in a minute. And importantly, the compound does not reach equilibrium with excess of compound solid. So this is quite important here, because that will affect the readout. But wait a moment. The compound concentration or precipitation can be measured by different methods. One is nephelometric assay. So here a series of dilutions is prepared, and we have a laser light scattering, and that detects whether there is a precipitation or not. This is quite good for automation and quite insensitive to impurities. Next, we have direct UV assay. So after short incubation, solids are removed and the concentration of the compound is measured by UV light absorption. It does not require serial dilutions. In the first case, we have these serial dilutions, and with the increased concentration of a compound, we finally will reach such a concentration that will cause precipitation, and that will be detected by this laser light scattering. Usually, because of all of this, kinetic solubility data can be higher than T-sol shake class method, because first of all, we add DMSO, which, as we described on the previous presentation, is very good solvent for almost everything. It needs to be in low quantity, but still we add a little bit of DMSO. Next, we don't have this equilibrium between solid state and dissolved state. Thus, the compound can actually be in a supersaturated state, because it takes time for them often to precipitate. Shaking the solution for 24 hours or longer can produce values that are comparable with T-sol data. But with this, we actually take out the pros, the advantage of this assay of being high throughput and fast. What are the pros and cons of kinetic solubility assay? So disadvantage is that supersaturation is often observed for low solubility compounds, and higher solubility values are being observed. If we apply the incubation time, then we may measure more reliable data. Often it is recommended to have the assay time that is similar to the time of the experiment that is being modeled. Then we will have the value that is informative for any assays that are of interest. But so why to select in general kinetic solubility assay if thermodynamic gives much better data, much more reliable data? First of all, compounds are readily available as DMSO stock solution. Companies have large databases, libraries of compounds as DMSO stock solutions to be readily available for any assay. So this is quite important because of the accessibility of molecules, of compounds. Next, the format of the assay is highly amenable for automation, so we can rapidly screen large collections of compounds. But I mentioned this on the previous slide, final DMSO concentration should be kept below 1% to not affect, not influence the readout. And solubility data can be time dependent. So with the varying length of the assay, we may measure different values of solubility. Importantly to remember, small differences in solubility data should not be overinterpreted, and this is because of this time dependency. The variability of metals, they generally increase at the lower end of the quantification range. Now let's move to the scheme for application of solubility assays. At the heat to lead stage of MedChem projects, we want to characterize the compounds based on the solubility, to identify whether the compound have a solubility viability, which means the best is take kinetic solubility assay because it's high throughput. If we want to optimize and develop a biological assay, the purpose is to have reliable IC50. We want to answer the question, is the assay reliable for low solubility compounds? The appropriate assay is a custom assay that will use the bioassay solution used in the biological assay that is under development. In the lead optimization stage, when we are optimizing solubility, we want to answer the question whether the structural modification improves solubility. It's okay to have kinetic or thermodynamic solubility assay. But in the lead optimization stage, when we want to diagnose poor pharmacokinetics, which means poor absorption, for instance, and is low solubility limiting absorption, the best is to have thermodynamic or custom assay. Custom meaning using some simulated biological fluids, because this uses the solid form that is actually being dosed to the animals. In animal dosing, we also want to answer the question, is the compound maximally solubilized for accurate pharmacology? And what is the best compound for in vivo study? Again, custom assay in various formulary components, which means in various solutions, various solvents that are being applied to dose the compound in this animal dosing. In the development of the clinical candidate, we want to know whether this compound meets the development selection criteria. Here, good choice is a custom thermodynamic assay in various aqueous formulary aqueous components that were required to be checked. And finally, in toxicity testing, we usually want to check what is the highest acceptable dose and what is the dose that causes toxic effects. So we would like to know whether we can obtain sufficiently high exposure levels for toxicity studies. And good selection in this case is a thermodynamic assay in these toxicity vehicles in vitro or in vivo. Now let's move to thermodynamic solubility example data. On the left, this is a representative table from one of the assays run at Cellvita. On the left, we have indicated what media were used for the assay. In this case, this is FASIF version 2. No worries, we'll explain this on the future presentations. Then we have number of compounds with their identifications, pH of samples after 24 hours, and actually outcome, results of the assay. Thermodynamic solubility at 37 degrees after 24 hours. Some compounds, as you can see, are quite poorly soluble, but some of them are very nicely soluble. 300. You remember 60 micrograms per milliliter was the benchmark for good or bad solubility. And compound number 7 is exceptionally well soluble. Some are very poorly soluble. And the next column constitutes comments. So scientists performing the assay will write down here all the observations, whether he or she observes some decomposition of a compound, any change of color, precipitation, anything that may be important for us to understand better the behavior of a compound in this medium. That's it for today, for this presentation. Thank you very much and see you next time. Bye bye.